behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, MatTek Corporation, I'd like to welcome you to Organotypic in vitro human respiratory models of the tracheobronchial and alveolar epithelium, EpiAir and EpiAlveolar. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. I'd like to introduce our presenters for today's webinar. First is Patrick Hayden, PhD, Vice President of Scientific Affairs for MatTek Corporation. Our second presenter is Courtney Menkes, PhD, Research Scientist on the EpiAirway, also with MatTek Corporation. And our third presenter is Rachel Gerlach, PhD candidate in the graduate program Microbiology and Immunology, Center for Predictive Medicine at the University of Louisville. With that, I will pass the presenter ball over to Patrick. Welcome, Patrick.
Okay, thank you, Elizabeth. I'd also like to welcome everyone to our webinar today. Our first slide shows an outline of what we'll be covering today. We do have a very diverse group today. Some of you are already familiar with MatTek, and some of you have prior experience with 3D culture technology. However, many participants may be unfamiliar or only somewhat familiar with the company and our products. So I'll begin with a brief presentation or brief introduction of MatTek and the basic technology that underlies our 3D human airway products. Then Dr. Courtney Menkes will present a survey of EpiAirway applications. And last but not least, Rachel Gerlach, PhD candidate from the University of Louisville, will present her work using EpiAirway to study influenza. Okay, before beginning, I'd like to take a moment to go over the learning objectives we hope you'll take away from this webinar today. First, you'll learn the advantages of 3D organotypic in vitro models derived from primary human cells compared to monolayer cells or Immortal cell lines. You'll also learn the basic features and applications of MatTek's organotypic in vitro human respiratory models. You'll learn what distinguishes MatTek's respiratory models apart from other commonly used 3D organotypic respiratory models. And finally, you'll learn how the use of EpiAirway or EpiAlveolar can accelerate your research. Okay, let's begin then with a brief introduction to the company and our technology. MatTek Corporation was established in 1985 in Ashland, Massachusetts, which is just outside of Boston. We produce a variety of in vitro models for toxicity testing, including skin, ocular, the airway, which is the main focus today, intestine, and other models. We have been the recipient of more than 30 small business SBIR grants funded by the NIH and the Department of Defense, and in fact, all of our products have been developed using NIH or DoD funding. So we've been very successful at transitioning grant funding into commercial products, and we're very appreciative of the support from NIH and Department of Defense. We distribute our products globally, and we have an extensive history of successful product utilization, as evidenced by more than 700 technical references that cite our products, as well as more than 400 U.S. patents that cite the use of MatTek tissues. This slide provides an overview of the basic process that we utilize to produce our products. This cartoon shows on the left, we see a cartoon of a human explant tissue, in this case, a cartoon of a human trachea, which I'll use as an example. So beginning with human explant tissue, we isolate a variety of cell types, and these could include epithelial cells, fibroblasts, other cell types as well, such as smooth muscles, etc., endothelial cells. We expand these cells in monolayer culture to produce a large pool of cells, which we then cryopreserve, so we maintain a very large pool of cells. And then as need be, we recover the cells from cryopreservation and utilize them to reproduce the organotypic cultures. This slide shows some of the features of the EpiAirway product. The top panel on the left shows an example of the H&E stained histology of the EpiAirway tissue compared to an in vivo human bronchial, which is shown. This shows the in vivo human bronchial in the lower section compared to the in vitro EpiAirway tissue. We can see the very similar structure and thickness, the so-called pseudostratified morphology of the tissues. Some of the main features of the EpiAirway product include a functional lifespan of more than 3 months. This allows repeat exposure experiments and long-term experiments. As I just mentioned, cultures have a pseudo-stratified morphology. The cultures have functional cilia. They secrete mucus. They have functional tight junctions, which provides them with barrier properties that are similar to in vivo. And they express appropriate drug metabolizing enzymes and drug transporters. This slide shows an example of the EpiAirway FT, or what we call the full-thickness EpiAirway, that consists of the pseudostratified epithelial layer, as well as a stromal matrix that contains viable fibroblasts. And here we see the microporous membrane, which is the support structure for the tissue. Okay, this slide introduces the EpiAlveolar. This is a new model that's currently under development. We expect to be launching it in Q4 of this year. This is a model, a co-culture of alveolar epithelial cells together with pulmonary endothelial cells. And this top panel shows the confocal, Z-stack confocal image showing the alveolar type 1 epithelial staining as shown by the green staining of keratin, cytokeratin 19, as well as the staining of the endothelial layer by von Willebrand factor, which is shown in red. And so we can see that these attributes are maintained for at least out to 30 days in culture. The lower panel shows an example of a top view showing the expression of tight junction proteins, in this case occludin, which is involved in producing the tight junctions, which provides the barrier for the tissues. So the intended applications for the EpiAlveolar will include general respiratory toxicology, nanoparticle toxicology, and lung permeation research, as well as tobacco, e-cigarette research, and general inhalation drug delivery research. All right, since some of you may not be familiar with the air-liquid interface culture systems, I want to spend a few moments just going over how this works. For those of you who are already familiar, this can be a refresher. This cartoon shows an example of a tissue culture insert, which is the basic feature of the platform. This tissue culture insert will have a microporous membrane, which is shown here. And then the tissues are cultured on the apical surface of the microporous membrane. The tissues are fed by adding culture medium in a well beneath the insert, so that the tissues are fed from below by percolating up through the microporous membrane. So media is never added to the apical surface. And this is very important for allowing realistic exposure scenarios. So we can apply chemicals or test agents, including smoke, aerosols, gas, or liquid materials directly to the apical surface in a more realistic fashion compared to submerged monolayer cultures, which are restricted to water-soluble materials. Here we see another pictures of the actual air-liquid interface tissue culture inserts. On the left panel, we see the individual insert. This just shows an example of a commercially available insert from Millipore Corporation. You may be familiar with other types of inserts as well, from Corning Transwells, for example. And they're available in a variety of sizes, including 6-well, 24-well, or 12-well sizes. And on the right panel is an example of high-throughput formats, including 24-well or even 96-well formats. Okay, then, so what are the main advantages of 3D organotypic models compared to monolayer cultures or immortalized cell lines? Perhaps the most important attribute, of course, is the differentiated structure and function. The 3D organotypic models will have in vivo-like barrier properties, cilia formation, mucus secretion, etc. As I already mentioned, the air-liquid interface is very important for allowing in vivo-like treatment. So we can model an apical exposure to environmental agents, cigarette smoke, air pollution, diesel exhaust, etc. Or we could even mimic systemic treatments by adding chemicals into the culture medium beneath the tissues. Our cultures also allow the study of individual variability. In our case, we never pool the cells together. We always use cells that are obtained from individuals without ever pooling them together. We prefer this method. I know some people tend to even use pooled donors. Well, we don't do that. In addition, we have donors who cells that are isolated from asthmatic donors or individuals who have had COPD or various smoking history. Regarding cell lines, I know there are a number of common cell lines, and a lot of people have done a lot of nice work using a variety of cell lines. We prefer, again, to not use cell lines. Mainly, we prefer to use normal human cells. So I guess by definition, the immortalized cell lines, just by the very fact that they're immortal, are not normal, especially if you're studying things related to proliferation or cancer, et cetera. And oftentimes, they may have deficits, defects in their function. For example, A549 cell line commonly used for alveolar doesn't have appropriate barrier. Another advantage of using primary human cells is that we avoid any species differences compared to using animal cells. A lot of people do use cells from some rats or dogs or other species. But with cells of human origin, there's no interspecies extrapolation needed. Of course, we can also better model human diseases such as asthma, COPD, smoking history, and also there are no ethical issues related to animal testing. What distinguishes MatTek's respiratory models apart from other commonly used respiratory models or models that many of you may be producing yourselves, homemade models in your own labs? We have a very long history of reliable commercial good manufacturing production. The EpiAirway model has been continuously available since the year 2000. The reproducibility and the quality of the tissues are guaranteed, so if there's a contamination or something goes wrong, then we will replace them, of course, free of charge. These cultures have a long lifespan. We can keep them going for repeat experiments for many weeks, out to even several months. We have a very large donor pool. Cultures, as I mentioned, always produced from single individuals. So that means that, since we have a very large pool of cells, you're able to use the same donor cells to do an experiment over and over again. So you can do an experiment tomorrow, you can repeat it 2 weeks from now, you could repeat it several months from now, or even in many cases, many years from now, because we do maintain our cell pools for a very long period of time. And of course, as I mentioned again, we have a variety of genders, ethnicities, smoking history, and diseases. Compared to producing cultures in-house, we feel that the cultures are very cost-effective. It allows you to focus your research efforts on doing your experiments rather than committing a large amount of time to culturing the tissues in-house. We feel it's a very cost-effective method, just obtaining the cultures from us. We have an extensive database of successful use by our customers, and we have excellent technical support and customer service. Okay, so that was really all I wanted to go over. I'm going to now turn the podium over to Dr. Courtney Mankus, and she's going to follow up with you and give you a survey of specific applications from the literature.
Thank you, Patrick. In this part of the webinar, I will introduce several applications in which EpiAirway can be employed to further your research. Today, we have a technical reference library of close to 100 publications that utilize EpiAirway. These consist of peer-reviewed publications as well as abstracts from conferences around the world that have been produced by EpiAirway customers as well as MatTek's team of scientists and collaborators. The complete list of the references can be found on our website, and from there you can also request full copies of any of the technical references. For each topic listed here, I will briefly review the technical references that are available in that category and then focus on one study in order to demonstrate the utility of EpiAirway. So the first application I will present is toxicology. Since EpiAirway is grown at the air-liquid interface, as Patrick described, it readily lends itself to toxicity studies in which the test article can be applied topically to the apical surface, which would be similar to the in vivo route of exposure through inhalation. In addition, you could also apply test articles in the basal media to replicate systemic exposure. An additional benefit is the ability to test non-water soluble substances, since for the apical application, you could dissolve your test article in non-aqueous vehicles. So there are currently 11 peer-reviewed publications in the toxicity category, examining anything from cigarette smoke and e-cigarette toxicity to airborne particulates, chemicals, pharmaceuticals, and consumer products. So in the category of toxicity, the study I'm going to be presenting is entitled Human Bronchial Epithelial Cells Exposed to In Vitro Cigarette Smoke at the Air-Liquid Interface Resemble Bronchial Epithelium from Human Smokers. And this study was published by Philip Morris in the APS Lung Cellular and Molecular Physiology Journal in 2013. So in this study, the EpiAirway tissues were exposed to cigarette smoke utilizing the Vitro smoke exposure system, which is shown here, with the goal being to determine if the biological changes observed in the airways of smokers in vivo could be reproduced in the EpiAirway model. The Vitro smoke exposure system consists of a smoking robot, which will actually smoke the cigarette. The cigarette smoke is then moved to the dilution chamber, where it is diluted with clean air to whatever the designated dilution is. The diluted smoke is then moved to the exposure chamber, which contains the EpiAirway tissues. So the tissues would be found in each of these wells that you see here. The smoke is exposed to the apical surface of the tissue through the aerosol inlet, which sits just above the apical surface of the tissues. So in this study, tissues were exposed to smoke for up to 28 minutes and then allowed to recover for up to 48 hours, during which at different time points the gene responses were analyzed. First, the responsiveness of the EpiAirway cultures to the cigarette smoke was determined by the measurement of pro-MMP release into the basal media. Pro-MMP release has been shown in previous studies to be upregulated in response to cigarette smoke, both in vitro and in vivo. So in this figure, you see that with an increase in exposure time, you have an increase in secretion of pro-MMP1, which indicates that the tissues are responding appropriately to the smoke exposure. Next, the investigators used gene set enrichment analysis to compare the in vivo smoking and smoking cessation gene signatures with the expression profiles of EpiAirway post-exposure. So here we focus on the in vivo smoking gene signatures and show 3 out of 4 gene sets that were analyzed in the paper. So each signature is separated into upregulated genes here on the left and downregulated genes on the right. And you can see that in all 3 gene sets, smoke-exposed EpiAirway tissues had significant normalized enrichment scores for 4 time points post-exposure for upregulated genes and at least 2 time points post-exposure for downregulated genes. So based on these results, the investigators concluded that EpiAirway tissues exposed to cigarette smoke are a valuable tool that can be used to further understand the biological impact of cigarette smoke on lung epithelium. So the next application that we will review is that of asthma and COPD disease research. As Patrick mentioned, we have a pool of approximately 30 donors, which vary in age, sex, smoking status, and disease status. Peer-reviewed studies that have performed thus far comparing these disease statuses look at histamine-induced airway remodeling, inflammation in asthmatic airways versus normal, and both intrinsic phenotypic and genotypic differences between asthmatic and non-diseased airways. So this study, phenotypic responses of asthmatic human airway epithelial cultures to rhinovirus and poly-IC, was performed by scientists here at MatTek in collaboration with collaborators at Merck. In it, the study was actually presented just last week at the ATS International Conference in San Diego. In this study, EpiAirway or monolayer cell cultures produced from 6 normal and 6 asthmatic donors were infected with poly-IC, which is a synthetic double-stranded RNA analog used to mimic viral infection, or live human rhinovirus. Baseline responses, as well as those following infection, were analyzed by Luminix for secreted cytokines and RNA sequencing for gene expression changes. So here you see on the left heat maps which show significant changes in either protein, cytokine secretion on the top, or gene expression on the bottom. Submerged cultures, which are just monolayer cells, are seen on the left, while the EpiAirway ALI cultures are on the right. So you can see that the submerged cultures were treated with poly-IC induced more significant changes than HRV in both cytokine secretion and gene expression, as indicated by the brighter red colors here under the poly-IC column. Alternatively, in the EpiAirway cultures, HRV induced more significant changes in both cytokine secretion and mRNA expression. So this suggests the importance of utilizing differentiated 3D cultures grown at the air-liquid interface together with HRV in order to better recapitulate the immune response to viral infection that occurs in vivo. The Venn diagram on the right shows differences both at baseline and upon HRV infection in the EpiAirway tissues produced from asthmatic and non-asthmatic donors. So you can see that in the baseline difference category, which is differences between asthmatic and non-disease donors without any treatment, there were 579 differences. The next category is the HRV response difference, which included 497 different differences that were asthma-specific viral responses. That is, changes that were seen only in the asthmatic and not in the normal tissues. And finally, the general HRV response category, which included strong expression changes associated with HRV infection in both asthmatic and normal tissues, included 1,485 changes. So for details on either the cytokine secretion changes or genetic differences observed in this study, you can go ahead to our website and request a copy of the poster, and we'll gladly send that to you. So the next topic I'll review is that of the use of EpiAirway for drug delivery studies. To date, we have 9 peer-reviewed publications focusing on nasal drug delivery, tracheobronchial permeation, tight junction modulation, and mucociliary transport. The study I will focus on in this category is the in vitro formulation optimization of intranasal galantamine leading to enhanced bioavailability and reduced emetic responses in vivo. And this was published by MatTek Pharmaceutical in the International Journal of Pharmaceutics in 2007. In this study, formulation of intranasal galantamine, which is used for the treatment of Alzheimer's disease, was successfully optimized using EpiAirway. They then followed up the in vitro experiments with bioavailability experiments using rats, and the pharmacokinetic test confirms an in vitro, in vivo correlation. When performing drug delivery studies using in vitro models, there are 3 things to evaluate. These are the disruption of TEER, or the trans-epithelial electrical resistance, which is an indication of tight junction functionality, tissue viability, and of course the actual permeation of the drug. That is, you want to be able to open up the tight junctions to allow for the drug to permeate into the tissue without reducing tissue viability. In this study, a pool of 3 permeation enhancers indicated in these results as PE was used to increase the transport of galantamine HBr across the EpiAirway tissue. In A, you see that the inclusion of PE significantly increased the permeation of the gal-HBr as opposed to the gal-HBr alone. Below that, in figure B, you see that the inclusion of the PE after 1 hour reduced the TEER. As I mentioned, this is an important property to allow for the permeation. The only other sample in the study that reduced the TEER was Triton X100, which was used as the positive control for tissue death. So as it was used to kill the tissue, obviously also induced a reduction in TEER. And finally, the 2 graphs on the right show 2 ways of measuring tissue viability, the top being MTT and the bottom LDH. And you can see that the inclusion of the permeation enhancers did not significantly affect tissue viability as opposed to Triton, which did. So as I mentioned before, this experiment was followed up by delivering the same combinations of drugs plus permeation enhancers intranasally to rats, and the results of these studies showed that the inclusion of the enhancers increased PK parameters and bioavailability. So therefore, you can see that the use of EpiAirway can provide an economical approach to initial screening and optimization of drug compounds. So the final application I'm going to present before passing the presentation over to our third speaker is that of viral and bacterial infection. There have been 16 peer-reviewed publications to date focusing on vocovirus, influenza, long-term host pathogen interactions, as well as antiviral and antibacterial studies. So the Danes lab from Mercer University has published 2 studies utilizing EpiAirway in extended co-cultures with H. influenzae. The first study, the use of EpiAirway models for characterizing long-term host pathogen interactions, is a video article published in Jove in 2011, and it actually provides very nicely protocols for unpackaging of the EpiAirway tissues, culturing of the tissues, and inoculation with bacteria. The follow-up publication, Characterization of Extended Co-Culture of Non-Typable Haemophilus influenzae with Primary Human Respiratory Tissues, was published the following year in 2012 in the Experimental Biology and Medicine Journal. And this publication goes into more details on the results from the extended co-culture. So under normal conditions, non-typable Haemophilus influenzae, or NTHI, is a commensal bacteria that becomes pathogenic at opportunistic times. So therefore, the study of how this bacteria interacts with respiratory epithelial cells is especially of interest. The graph on the top left here shows recovery of the NTHI from the co-culture of EpiAirway for up to 10 days post-inoculation. So this, you can see that even after 10 days, the bacteria are still maintained within the EpiAirway culture and can be recovered. The EM images on the right show the apical surfaces of the EpiAirway tissues that have either been infected with NTHI or left uninfected. And you can see that after 5 days of co-culture, the apical surface is not significantly damaged compared to control tissues. Therefore, bacteria can be co-cultured with the EpiAirway tissues without detrimental effects to the tissue ultrastructure. And finally, the EM images on the bottom left show after 5 days of inoculation, the NTHI is mostly paracellular, as indicated in the red arrows here in A. The boxed area here in A is blown up larger in B and shows bacteria that has been found intracellularly and is shown as being degraded. So you can see that EpiAirway is able to be maintained in culture long-term in co-culture with bacteria for up to 10 days and most likely even longer. So in addition to bacterial studies, EpiAirway has also been used extensively to study viral infections. At this time, I'd like to pass the presentation over to Rachel Gerlach, a PhD candidate at the University of Louisville, who will present her work utilizing EpiAirway for influenza studies.
Thank you, Courtney. Very thankful for the invitation from MatTek to present the research done in my lab using the EpiAirway tissues. This work was done in my laboratory at the University of Louisville. We study virus-host interactions, specifically influenza virus. I'll begin my talk with a brief background of influenza virus and highlight the importance of studying these viruses. I will then briefly explain why we chose to use the EpiAirway model over other in vitro models, and will present data from a recent publication from our laboratory using this model system. So briefly, influenza virus is a respiratory virus. It infects roughly 500,000 people in the United States each year. Despite yearly vaccine campaigns by the CDC, influenza epidemics and pandemics create substantial morbidity and mortality. One of the worst pandemic outbreaks to date, the 1918 influenza, caused roughly 1 million deaths in the United States and 50 million worldwide. Each year, even with more mild influenza epidemics, the economic impacts are substantial, with billions of dollars being spent on direct medical costs and lost earnings due to illness or loss of life. Unfortunately, there are limited treatments available for influenza infection, and drug resistance is becoming apparent with drugs on the market today. The need for improved treatments is a necessity. So most of us know the symptoms of influenza all too well. This includes fever, sore throat, body aches, and cough. The symptoms show up 1 to 4 days after you're exposed to the virus, and this virus can be transmitted either by the respiratory route, so drinking directly from someone, or indirectly touching contaminated surfaces and then touching your eyes, nose, and mouth. So how does the virus infect and cause disease? Influenza primarily targets the lung epithelium. This includes both the upper and lower respiratory tract. Most yearly and seasonal influenza epidemics are caused by viruses that infect the upper respiratory tract. while pandemic H1N1 viruses, such as the one that we saw in 2009, infect the middle airways, such as the bronchioles. Highly pathogenic H5N1, also known as avian influenza, which is fortunately very limited in our global population so far, this is seen to infect the lower airways, or alveoli. The severity of disease has been shown to correlate with the location of infection. So the cells that are present in different sections of the lung are heterogeneous, with the upper airways containing most ciliated and mucus-producing cells, and lower airways of cuboidal cells that are highly responsive to bacterial and viral stress. Once the virus gets into the lung, how does it infect? So influenza virus infection is initiated by the binding of surface protein, the hemagglutinin, to its receptor, a galactose-bound sialic acid on the host cell surface. Once bound tightly, the virus undergoes endocytosis, and upon acidification of that endosome, the viral components are released into the cytoplasm. At this point, the host can recognize the virus and respond via several intracellular signaling pathways, such as toll-like receptor and RIG-I, that's signaled through NF-kappa B. The virus genetic material then gets shuttled into the nucleus where the virus and host machinery create the viral RNA. This is then followed by protein synthesis of the necessary proteins that are required for the virus to replicate its own genome back inside the nucleus. Once all the viral components are made inside the cell, they are shuttled to the cell membrane where progeny viruses are formed, and budding and release is initiated by this neuraminidase protein on the virus surface. That newly synthesized virus can further infect other cells and can be transmitted through coughing or sneezing. In order for the virus to infect epithelial cells, there are two necessary components: receptors and also proteases. Sialic acid receptors specifically targeted by influenza virus, and the proteases produced by the lung epithelial cells. The absence or presence of both is critical to influenza pathogenesis. Attachment of all influenza strains to cells requires sialic acid receptors. These receptors can take on different forms chemically, depending on the binding orientation of galactose to sialic acid on the cell surface. Influenza virus strains can vary in their affinity for these different conformations, and these differences can influence the location of the human lung that can be infected. Most widely reported orientations favored by influenza HA include the alpha-2,6 and also alpha-2,3 sialic acids. Both can be found in the upper airway, as seen here, but as you move further down the lung, the receptors become predominantly alpha-2,3. This becomes a very important factor when studying influenza because specific affinity can influence disease severity, which is often associated with lower respiratory pneumonia. The images over here on the right demonstrate both of the receptors are present on the EpiAirway differentiated model. Another important determinant of virus infection are the presence of host cell proteases, which are necessary for the activation and entry of influenza virus. As you can see here, H0 represents hemagglutinin protein that has not undergone a conformational change. In order for the virus to bind and infect the epithelial cell, the HA must be cleaved to expose the fusion peptide domain, but also exposes the sialic acid binding domain, allowing the cell to attach and also enter the cell. If none of these proteases are available for the virus cannot bind, and it cannot infect the susceptible epithelial cell. Our lungs contain several proteases, as seen here by these top three, and are important factors to consider when choosing a model to study pathogenesis. In this report, down here, differentiated cells were shown to have a greater than 700-fold expression of host cell proteases compared to an undifferentiated model. This further highlights the importance of using a differentiated model when studying influenza. When I joined the laboratory at the University of Louisville, the first question we had was whether we could discriminate between seasonal and pandemic 2009 influenza isolates using epithelial models. The panel of influenza isolates our laboratory is using were obtained from hospitalized patients as a part of an ongoing severe influenza pneumonia surveillance program in the state of Kentucky. I'll draw your attention to Kentucky 180 and Kentucky 136. These two isolates were isolated from Kentucky in cases where a person died, Kentucky 180, and Kentucky 136 where the patient survived. These isolates also showed the very same phenotype in mice. So we asked whether we could discriminate between these isolates using an in vitro lung epithelial model. Our approach to address this question was to select different epithelial models found in the literature and see if these differences occur in infectability. We chose 4 different models, 2 continuous cell lines, CALU3 and A549, and 2 primary epithelial models representing undifferentiated, a monolayer, and then a differentiated epithelial multilayer. We first wanted to know how well our virus is infected and replicated in each cell type. To measure this, our lab performs what's known as the TCID50 assay, and the numbers are indicated on the left y-axis. That measures the amount of virus that is released from the cells over time, so you see our time points on the x-axis. The most significant difference in replication among isolates was seen in the human primary lung epithelial models, the undifferentiated and well-differentiated models, but to a greater degree in the differentiated EpiAirway model. Significantly, Kentucky 180 showed a 1 to 2 log greater level of replication across the time course of infection. And these results suggest the relevance of using this model to understand early differences between isolates in the same strain. So why use this model? Well, this model has been demonstrated to contain both receptors necessary for influenza infection. We've also shown that our viruses, and along with others, can productively infect and replicate in this model. This model is more physiologically relevant than the other 2 in vitro models we tried because they do not have to have added endogenous proteases to those cell culture media to get infection. And finally, because these EpiAirway tissues are at the air-liquid interface, we're able to see what the signals were produced by infected epithelium, both apically and basally. This provides a more comprehensive look at what the virus is doing to the epithelium and the microenvironment established around the cells. So ultimately, this model was chosen because it closely mimics the lung, the human lung, that the virus would encounter naturally. Our next question was, could we use the EpiAirway model to further understand early host responses to seasonal and pandemic influenza infection? We used the EpiAirway tissues from MatTek, and we repeated this experiment 3 times. The cells were infected with our isolates at 1 virus per cell at 8, 24, 36, and 72 hours after infection. We collected cellular supernatant, both apical and basal, and further collected the cells using a reagent to isolate RNA, DNA, and protein, or fix the cells for immunohistochemistry. To evaluate the cells by immunohistochemistry, we made paraffin-embedded blocks of the cells in counterstate with algae in blue and influenza and protein seen in brown. These representative sections are shown for each virus and an uninfected mock control. The pictures here show cross-sections of the epithelial layer with apical and basal side, as you can see here, the apical and basal. One of the known results of influenza infection is desquamation of epithelial cells, and this was more pronounced in Kentucky 180 infected tissues. To confirm this observation, we used imaging software to measure the distance between the membrane and the top of the epithelial layer, as you can see here. What we found was a significant difference between the lethal isolate and the mock infected control and other isolates in terms of thickness of the epithelial layer, as Kentucky 180 represented in red. Further, we looked at the expression of genes that would normally be seen in a differentiated phenotype. This includes genes that are involved in celiogenesis and mucus production. We saw a greater change in expression in those genes in Kentucky 180 infected cells, our lethal isolate, compared to our non-lethal and seasonal influenza. Our next objective was to use microarray to evaluate the gene expression patterns in infected tissues and evaluate whether we could pinpoint differences between viruses to specific intracellular pathways. The microarray data from the isolates were normalized to mock-infected well and filter using a 2-fold and a p-value of less than 0.05 cutoff. Interestingly, we saw very few differences between our Kentucky isolates. However, we did see clear differences in pandemic expression compared to seasonal, reflected by the regression plot here on the left. Using a heat map to visualize the expression levels, and this is represented by fold change of secreted cytokine, chemokine, transcription factors, etc. here, we show significant similarity in response across all pathways between our 2 endemic isolates. However, they did differ from seasonal. Our final objective was to determine whether differences existed in cytokines and chemokines secreted from infected EpiAirway tissues. After infection at the designated time points, the apical side was washed and evaluated for secretion of 12 cytokines and chemokines at the 5 time points. The time points are here on the x-axis, and then the concentration represented in picograms per mil on the y-axis. This was done using a multiplex bead assay, and the data I'm about to show you is representative of 2 independent experiments. As shown here from the apical wash, 8 of the 12 cytokines and chemokines showed very similar trajectories, the ones not highlighted in yellow. And this was done for all 3 isolates over the time course of infection. For those highlighted, CCL5, TNF-alpha, interferon-alpha, and CCL2, we observed unique profiles for each isolate, seasonal and pandemic. When comparing the lethal and non-lethal isolate from the apical wash, we found that CCL5 was the only discriminator between the two isolates. As was done for the apical side, we evaluated the secretion of 12 cytokines and chemokines using the same magnetic bead assay. From the basal side, we saw a greater discrimination between the three isolates with those ones that are highlighted in yellow. When comparing our lethal and non-lethal isolate in the basal supernatant, we found that CCL5, CCL2, IL-6, and MIP-1 beta were secreted to significantly greater levels across the different time points. Overall, the lethal isolate, Kentucky 180, produced greater levels of infectious virus across the time ports. Greater levels of IL-6, CCL2, CCL5, and MIP-1 beta were secreted by Kentucky 180 infected cells, with a greater signal coming from the basal side. However, the gene expression profiles do not correlate with those differences in secreted cytokines and chemokines. From this data, we hypothesized that the differential basal secretion would differentially affect monocyte recruitment and migration, and the signals from infected EpiAirway tissues would promote an increased macrophage signal and further modulate the immune response. Had we not used this model, we would not have seen differences in basal secretion, which led us to look at more downstream immune cells that are recruited to the site of infection. I will end my presentation with our laboratory's working model of influenza infection with viruses of the same strain. Our next objective is to see how the viruses alter or interact with recruited immune cells, and then further look at interactions between EpiAirway tissues and those recruited cells. This co-culture would further demonstrate how microenvironment established by EpiAirway cells could influence pathogenesis. Thank you very much for your time and attention, and I will turn it back over to Elizabeth.
Thank you very much, Rachel. We've had quite a stream of questions coming in, so let's get started and get to as many of them as we can. The first question that we have is, I have experience producing ALI cultures in my lab. What are the advantages of using EpiAirway?
Okay, thank you, Elizabeth. This is Patrick. I'll go ahead and address that. I think the main advantage of using the EpiAirway is our good manufacturing production process that provides the reproducibility of the tissue. We test all our supplements and all the cells, and everything is pre-tested, so you're guaranteed reproducible product week after week after week. The quality of the tissues are guaranteed, so if there is a contamination or for whatever reason the tissue doesn't pass the QC specs, the customer doesn't have to pay for it. So perhaps the other, maybe even a bigger reason for purchasing the tissues pre-made from MatTek is the extensive time commitment that's required for people to make their own in-house models. I know a lot of people do make their models. Some people can make really nice models. Some people have trouble with it. But in any case, it's a significant time commitment. And so we just ask people, do you want to spend all of your time in the lab culturing these models, or do you want to spend time doing your research? So to purchase them from us is just very convenient and economical. Show up on your doorstep, you recover them overnight, you're ready to go with your research. Again, the models are very well characterized, and we have a good donor inventory with a large pool, so you can use the same donor again and again, especially now with our asthma donors, for example. We've already got them characterized. The work is already published. So we know there are differences between the asthmatic and the non-asthmatic, and now you can use these same donors in your own research. So you don't have to go back and reestablish that all over again. Again, I think we think that our models are actually a very cost-effective means of doing research with ALI tissues.
We've got questions coming in so fast I'm having trouble keeping up with them, but I'll do my best. How does data produced with EpiAirway correlate with data produced from in vivo systems?
So there are a number of published reports from EpiAirway users that have provided some very promising examples of in vivo correlations, including the prediction of nasal drug delivery and irritation. For example, the publication from MatTek that Courtney presented. The cigarette toxicity work that's been done by Philip Morris and others also showed a nice correlation to what has been previously obtained in vivo. The asthma work that we did on our own in collaboration with Merck and academic collaborators has shown the expected response of the asthmatic tissues. It's very similar to what's been observed in vivo. There has been, in fact, just this morning, a patent application from CTOX, which is a CRO that utilizes EpiAirway. They published a patent application for predicting airway toxicity in EpiAirway. So what we recommend for people when trying to do toxicity studies is to always use a benchmark chemical, something that they know well in their own hands. This is irritating in vitro or in vivo or not, and then they can benchmark their new formulations against that. So I think that tends to work well for people.
Okay. We've had several questions come in, and I kind of compressed it into one question. How long can the tissues be maintained in culture, and do you have specific protocols for extended culture?
We have kept our cultures going for out to 3 months and probably could have gone longer. You do have to take care of them. We feed them every other day. We can provide a protocol for feeding with the volume of medium, which may depend upon the exact format that you get. We provide numerous size formats, including 96 well, 24 well high throughputs as an individual well. So we can provide that to people. The feeding schedule, maybe once a week or once or twice a week, rinse the apical surface off with some PBS. And if you take good care of them, they can definitely go for weeks to out to at least several months.
All right. Have you done any research on respiratory irritation caused by inhaled aerosols? And if so, what co-culture would you expect to be better for this purpose?
Okay, well, there's been, mostly in our own hands, we've done cigarette smoke work. And regarding the tracheobronchial versus the alveolar, I'm not sure which one is necessarily better. And the alveolar is a very new model, so we don't have a real strong history and background with it. But certainly it's going to be a little more sensitive, or significantly more sensitive. It's a thinner tissue, it's a little bit more fragile. But I think both models could be, useful for doing toxicity and irritation studies. As far as exposures, I mean, people have done, you need a special apparatus for cigarette smoke, or if you want to expose them to various aerosols, like diesel exhaust or some kind of drug aerosol. That can certainly be done, and I would again recommend to benchmark it against a known irritant formulation that's similar before you test your unknown.
Related to that same question, what endpoints can be used when you're looking at respiratory irritation?
Okay, there are all kinds of endpoints that can be used. You can look at cytokine secretion into the medium. It's commonly IL-8, IL-6, a whole variety of cytokines. Tissue viability is maybe even the best indicator of irritation, and this can be done by things like the MTT assay, measuring LDH or ATP. The barrier function can be measured by measuring transepithelial electrical resistance. Ion channel functions can be measured. Just general histology can be looked at, the H&E stained paraffin sections. Immunohistochemistry can be used to look at the localization and expression of specific proteins. Gene expression by RT-PCR or microarrays or RNA-seq can be used. Western blotting, I mean, virtually anything you could do to a piece of tissue in vivo or even cells in vitro, you can do similar endpoints with the EpiAirway.
All right. A person asks, have you had a chance to do any preliminary studies to test nanomaterial toxicity in the EpiAlveolar model?
We have not done anything to date with the EpiAlveolar. It's still very new. We did do some limited work, and some of other users have in the past done some nanoparticle work with the standard EpiAirway tracheobronchial model, using things like polystyrene nanoparticles. We've done, I guess people have done carbon nanotubes, et cetera. So I do expect more work to be done in that area. We do feel like that's going to be one of the big applications for EpiAlveolar, but to date, it's so new that we haven't done much with it yet.
All right. Keith asks, can we add plasma or serum or human serum albumin to the incubation matrix to to mimic blood compartments?
Well, we have not done that. I think it could be tried. The only caveat might be that I'm not certain what the long-term effect of adding all that plasma, all the serum growth factors that would be present. I'm not 100% certain on how that might alter the tissue long-term.
Can this model be used in genetic toxicology?
Absolutely. There are a number of toxicology assays that would probably be applicable to this. We can measure DNA damage by the H2AX marker. You can measure P53 activation. We've also done COMET assay and also the micronucleus assay. So, there are all kinds of genotoxicity assays that you could bring to bear with the EpiAirway model to look at cigarette smoke or whatever.
All right, we've just come up on the hour, so one more question. Can you use your system to mimic hypoxia?
Good question. We've never tried that, but certainly you could, if you have an incubator where you can, or a chamber that you can control the oxygen concentration, then yeah, absolutely that can be done.
Okay, perfect. Well, thank you so much, Patrick, Courtney, and Rachel, for your presentations today. I'd like to thank MatTek Corporation for sponsoring. Most of all, I'd like to thank those of you who came and spent some time with us to learn about this technology and perhaps find some answers that will help you in your research. So with that, I'd like to say thank you so much for coming. I look forward to seeing you at future CHI Global Web Symposia, and I wish you all a good day.
Thank you, Elizabeth, and thank you, Rachel, as well, and to all the attendees. Goodbye.